Welcome to my channel Adhyan with Flow. Let's start with endomembrane system. This endomembrane system is made up with ER, endoplasmic reticulum, and Golgi body. Okay, we're going to study about ER plus Golgi body, and overall we call it as a endomembrane system. Because guys, this endomembrane system they give their huge contribution in protein sorting. Yes guys, protein sorting. And now what is protein sorting? They produce, they transfer message one organelle to another organelle. How? This is the guy nucleus. Assume now this is the nucleus. Here is ER will attach, and here Golgi body. Clear guys? So nucleus contains genetic material DNA. DNA replicate and DNA will form RNA. RNA form protein, and this protein they do as a messenger work. They transfer nucleus to ER, and transfer into Golgi body, and they transfer into peroxisome, they transfer into plasma membrane. Okay, now let's start one by one. Look at here guys. This is the nucleus guys. Look at here. This is the nucleus. This is the nucleus. And here is nucleus pore. So message can easily come out with this pore. Message will come out easily with this pore. And what are these? What are these? They are endoplasmic reticulum. What are these? These are endoplasmic reticulum. Clear guys? Now very clear. Now there is two types of ER is present. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum guys. Yes guys. What is the function of rough endoplasmic reticulum? Of course they carry function. Of course they carry message. Of course they do their function. Okay, we will study. But why we say rough ER? Because these ER contain ribosomes, and we already discussed about ribosome. What is the function of ribosome? They perform photosynthesis. And some ER, these are smooth ER. Why we call it as a smooth ER? Because it does not contain any kind of ribosome. So they perform their function as a storage house. Alright, so let's start reading NCERT. Electron microscope studies of eukaryotic cell reveal the presence of network of reticulum of the teeny tubular structure scattered in the cytoplasm is called as a ER. They said that they scattered in their cytoplasm attached with nucleus. ER divide the intracellular space into two distinct compartments, luminar and external. This inside is connected with inside ER, and external, these part is connected with directly with cytoplasm. Okay, when ribosome attach with ER, we call it as a rough ER, and if there is no ribosome, then we can call it as a smooth ER. Ribosome is frequently observed in the cell actively involve the protein synthesis. Of course, when ribosome is present, then you have to clear that they are responsible for protein synthesis. The smooth endoplasmic reticulum is a major site for synthesis of lipid. You know now our lipid, our plasma membrane is made of both phospholipid bilayer. In animals, a lipid like steroid hormone are synthesized in SER. They also, what they do? They synthesize what? Lipid and some steroid hormone. Clear guys? Very clear. Now let's talk about Golgi apparatus. We can also say it as a Golgi body. Okay, so Golgi apparatus is just behind the ER. Like this, like this, like this. This is the cis median and trans, and this is the what? This is the lysosome, and there is plasma membrane. So here is ER, they attach with nucleus. ER get message from the nucleus. ER provide message to Golgi body, and Golgi, how they transfer their message in the form of vesicles. They code protein, and this vesicle reach to the next Golgi body, and this message vesicles accepted by median Golgi, and they transfer their message in the form of vesicles to another. Here is your vesicle is reached, and they transfer their message in the vesicle form, and they reach to lysosome, and lysosome make a vesicles, and this message is reached toward the plasma membrane. Can I repeat again? Can I repeat again? This is the nucleus, this is the ER, this is the ER guys, and this is the whole Golgi complex. Now what happened? Nucleus provides some signal, gives some message to ER. ER code this message and transfer this message towards the cis Golgi complex. Cis Golgi complex accept this signal, but they transfer their message towards the median in the form of vesicles. And this vesicles accepted by median, and this median code their message and they transfer their message towards the trans in the form of vesicles, and this vesicle is accepted by the trans, and trans gives their message towards the lysosome, and lysosome will receive the message and code to according to their cell, and they provide message to the plasma membrane. Guys, this is the endocrine system. Guys, this is the endocrine system. Guys, this whole is endocrine system. Means here is the game of message. Nucleus codes a message, they provide towards the ER provides message to Golgi apparatus. Golgi apparatus made up with three parts: cis, median, and trans. This is the old and this is the new forming. This is the new median trans. The message in the vesicle form, and they provide message to lysosome. Lysosome provide message to PM. This is the endocrine system, endomembrane system. So, who discovered Golgi body? Camillo Golgi observed densely stained reticular structure near the nucleus. This is the nucleus. Here is ER and here is Golgi. These were later named Golgi bodies after him. They consist of many flat disc-shaped cisternae. Look at here size. These are stacked parallel to each other. Yes, guys, parallel to each other. Varied number cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct concave cis forming. Guys, look at here now arrangement. Look at here their arrangement. Parallelly arranged near the nucleus, so they can easily get message from the nucleus. Golgi apparatus principally perform the function of packaging material. I told you na guys, protein, sorting. That's it. This is the simple word and this is the scientific word. To be delivered, look at here guys, to be delivered either to the intracellular target or secreted outside the cell. I already described na guys. I already described this thing. They secreted their message to their target organ. They secreted their message to their target organ. They secreted their message to their target organ. Material to be packaged in the form of vesicles. Now you clear guys? They give message, not directly. They give message in the packet form. We call it as a vesicles. From ER fused with the cis phase. Look at here guys. This is the ER and this is the cis. This is the median. This is the trans. Where I am from ER fused with cis phase to Golgi apparatus and move towards the mature phase. This is the new median and trans. This is the mature and this is the newly formed Golgi apparatus. Why the Golgi apparatus remain in close association with the endoplasmic reticulum? A number of proteins synthesized by ribosome on the endoplasmic reticulum. Yes, we know na that ER also contain ribosome. We call it as a rough ER. Rough ER. Why we say rough ER? Because they contain ribosome. And what is the function of ribosome? Protein. And protein have multiple message for all cell organelles. So that's why Golgi attach with ER. So they directly grab their message. Golgi apparatus is important site of formation of glycoprotein and glycolipid. ER also contribute to formation of lipid, but Golgi apparatus they helpful in formation of glycoprotein and glycolipid. Carbohydrate plus protein, carbohydrate plus lipid. Sugar part na guys, this is the sugar part plus protein, sugar part plus lipid. Okay, now what is lysosome guys? What is lysosome? Now let's study about lysosome. We can call it as the suicidal bag also because they contain highest acidic enzymes like lipases, proteases, carbohydrates. Lipases they are break down lipid molecule. Proteases they break down protein molecule. Carbohydrates they break down carbohydrates guys. Now you clear? Let's study about lysosome. What are the lysosome? This is the membrane bound structure. Which kind of membrane? Simple guys now, plasma membrane guys. And acidic volume is very high. Acidic nature means their pH is 1, 2, 3, 4. They contain lots of hydrolytic enzymes. Like they contain enzymes for lipid breakdown, they contain enzymes for protein breakdown, they have enzymes for carbohydrate breakdown guys. Lysosome arises from Golgi body. They are arises from Golgi body. So guys, that's all for now guys. That's all for now. In our next video, we will start with vacuole and mitochondria. So thank you guys. Thank you for listening me and please subscribe my channel. Keep smiling. Thank you.